Okay, so last we talked about the first half of meiosis, meiosis 1, and remember meiosis 1 is all about moving the homologous chromosomes away from each other. So now we're moving on to meiosis 2. So this second half, this second cell division. And in meiosis 2, what we're separating is the sister chromatids, the ones that are attached by those centromeres. Okay, so this would be meiosis 2. That's what we're going to focus on today. And remember, meiosis 2 is just a repeat of the stages of meiosis 1. They have the same names. And again, remember PMAT to help you remember the order. So it would be prophase 2, metaphase 2, anaphase 2, telophase 2. So let's take a look at what those look like. So remember, prophase 1 is all about the nuclear membranes breaking down. You're going to get those centrioles moving to either end of the pole. Prophase 2 is also where you start to see those spindle fibers coming off the centrioles and start attaching to the chromosomes on their kinetochores. Remember, the kinetochore is what hooks up the spindle fiber to the centriole or on the chromosome, and the centromere is the protein that holds the sister chromatids together. Okay, so that's it for prophase 2. Nothing else really important happens there. The other way that you would recognize prophase 2 is if there were, disorganized chromosomes, like you couldn't really see them, the linear shape, but there were two cells. That's how you would distinguish it between prophase 1 and prophase 2. Again, prophase 2 does not have any crossing over. That only happens in prophase 1 between the homologous chromosomes. So in prophase 2, there is no crossing over. Okay, so after you have your spindle fibers attached in metaphase 2, remember that stands for middle. So the chromosomes are going to align along the center of the cell, along that central axis. And again, this is metaphase 2 because you'll be able to see two of them. And again, which pole each sister chromatid faces is random. So in this example, the darker chromosomes are on the bottom side, but either one could easily be flipped upside down. So after they are aligned along the middle and the spindle fibers are now attached, these guys are ready to start moving. So again, in anaphase is when the chromosomes are moving away from each other. So in anaphase 2, it's the sister chromatids that are being fully separated and pulled into these opposite directions. And again, in telophase, that's when you can start to see the formation of the four cells. So in anaphase, you would see them moving away from each other, and in telophase, if you can see four different cells, that's telophase 2. And again, this is just like the cleavage furrows before they're fully formed. That's what you would start to see the four different cells. Okay. So this is the whole overview of both interphase, where the chromosomes are replicating, and you will have to be able at some point to draw these and label them and tell me what's going on in each one. Okay, so this is the 90-second recap of the stages of meiosis. So remember that we start first with the replication of the chromosomes before meiosis can even happen, because we need to make those four haploid nuclei, and we're starting with a diploid cell. So the way that we do that is we first have to replicate those chromosomes in the S phase of interphase. So that is where we get the creation of the sister chromatids. Those genetically identical sister chromatids happen in S phase of interphase. So after that, now we can start meiosis. So remember that prophase 1 is where you first have the condensing of the chromosomes, and something else really important in prophase 1 happens, and that is the exchange of alleles in homologous chromosomes, known as crossovers. So these are two homologous chromosomes, and what they're going to do is swap sections, creating totally new chromosomes. So you see this one here, and it's going to swap a piece. Remember that homologous chromosomes are similar, but not identical. They have different alleles. So what this is doing is it's creating an entirely new chromosome, a new set of alleles that did not exist before over here and back here. So this is just a way to add genetic variety. So after those crossovers happen in prophase 1, the chromosomes are going to align in the middle of the cell, and you know that it's metaphase 1 because we only have one cell that's visible. And So they'll line up there. And again, remember that orientation is random. So who's on the left side and who's on the right can go either way. This is going to give you even more variety. So you have basically two to four possibilities for every set of chromosomes. So that's a lot of different possibilities in metaphase 1. So after they line up and the spindle fibers are attached, now the chromosomes can move away from each other. So this is anaphase 1, when the chromosomes are moving away from one another. And in telophase is when you can start to see the separation and division of those cells. So you know that this is telophase 1 because there are only 2 visible cells. Telophase 2 would have 4 of them. So you know this is telophase 1 because there's only 2. It's the first cell division. So after that, the whole process starts over again. So in prophase 2, again, the nuclear membrane is going to break down. You're going to get a condensation of those initial chromosomes. They become very visible. So no crossovers happen in prophase 2. Crossovers are only between homologous chromosomes in prophase 1. So in prophase 2, all you're seeing is the condensation of these chromosomes in 2 different cells. So that would be prophase 2. So in metaphase 2, they're going to once again align in the middle, and you know it's metaphase 2 because there's 2 different cells. So, and again, the chromosomes align along the center. And once again, the orientation is random. So it could easily have been that this chromosome is going to be on that side, or that chromosome would be on this side, and that just gives you more variety in the ending haploids. So, okay, they line up in the middle for metaphase. And then again, in anaphase is when they're going to start to pull apart. And then telophase is when you can see the different cells. And in telophase 2, you have 4. Okay. So those are the stages of meiosis. Okay. Back over here. So this chart is just meant to help you be able to tell them apart. So go ahead and fill this out. So we start with S phase of interphase, and that's when the duplication of chromosomes to create sister chromatids takes place. Again, you can only see one cell because they haven't divided yet. So prophase 1 is when those chromosomes become visible, and you have those crossovers between homologous chromosomes. And again, everything until you get to telophase 1, it's just one cell that's visible in meiosis 1. Metaphase is when the chromosomes align at the center. And anaphase is when those chromosomes start moving away from each other. And again, remember from meiosis 1, what is primarily being acted on are the homologous chromosomes. And in telophase 1, you get those new cell membranes forming, and then you can now see 2 cells. So when we move to that second division in meiosis 2, from here on out, how you distinguish is that there are 2 cells visible. And again, in meiosis 2, what is being separated are the sister chromatids. So in prophase 1, all that's really happening is that the chromosomes become visible, they condense. And again, in metaphase 1, these sister chromatids then start to align along the center. And in anaphase 2, sorry, this should be all 2. So prophase 2, metaphase 2, anaphase 2. I hope it didn't print like that. So on your sheet, if you see this, just add the 2s so you know that it's a different, we're in meiosis 2 now. So in anaphase 2, it's the sister chromatids that are moving away from each other towards the opposite poles, and in telophase 2, that's when you have the formation of the 4 visible cells. Okay, so if I ever ask you to draw these, please be sure that you're indicating at least, 2 sets of chromosomes, indicate your spindle fibers correctly, label each stage